thank you everybody for coming. I guess I'll just dive in. I'll ask you to keep yourself muted just for static reasons if you're not asking a question. But if you have a question, please feel free to unmute and ask. And yeah, I guess let's dive in. My name is Mikhail Tikhonov. I'm an assistant professor here at Washington University in physics. And let me can you see my slides now, or do you see something else? Okay, yes. Excellent, perfect, wonderful. And hopefully you can also see a laser pointer.
Yes.
Okay, wonderful, thank you. All right, yes, so the title of my talk today is The Next Revolution in Physics, Microbial Ecology with a question mark. And as you probably noticed, this title makes no sense because if the next, maybe there's exciting things happening in microbial ecology, but what does it have to do with physics, and especially theoretical physics? So my hope today, nevertheless, is to try and argue that I believe that the next revolution in theoretical physics will focus on the living world and will transform the very language we use to describe it. Now, obviously, I can't know that. Nobody can. And in science, we always use kind of might and could, you know, and maybe instead of will. But today I'll put on my speculative hat and you know, tell you about the dream that I'm excited about and that motivates the science that I do. And that's my plan for today. But the glaring question that I sort of need to address first is, okay, focus on the living world, but we already have a science for the living world, and that's called biology, and biology is not physics. So what is physics doing here? And the story goes something like this. Theoretical physics as a discipline has been incredibly successful. And as a result, theoretical physicists have become incredibly arrogant, and they barge into other disciplines thinking they can improve and revolutionize and change economics, sociology, biology, etc. Okay, so I'm joking, but I sort of, well, I wish I could tell you this is just a joke. Sadly and unfortunately, physicists do have a bit of a reputation of talking about things that aren't physics as if they know what they're talking about. So let me try again. And my hope today is to convince you that my story today is not this. Okay, so let me try again. Theoretical physics has been incredibly successful. And that is my starting point. And in fact, theoretical physics went through a sequence of revolutions. And as we witnessed that process, we learned something. Because over and over, we had to revise some of our most fundamental certainties, things that we believed couldn't possibly be any other way, transforming how we view the universe today compared to, you know, a couple hundred years ago. And through that process, we learned to spot the clues that you know, suggest that there might be something interesting here and something big. And so I'll argue to you today that the microbial universe, as we're learning, what we're learning about microbes is setting off these alarm bells and make it a very exciting subject for theoretical physicists to think about. And so in the story that I will tell you, the relationship between physics and biology looks something like this. So there's a big enterprise called biology, and there's also a big enterprise called physics, and physics does something that they're interested in, but they can also contribute something to biology. And what can they contribute? Well, they can contribute tools, like here's this ultra-fast measurement device built on lasers, right? Or this single molecule precision jackhammer, right? Physicists can build tools, But also there's kind of this guy who's been, that's the kind of my depiction of a theoretical physicist, who's been trained as a kind of treasure hunter. And this guy says, don't mind me, but you know, like this thing here, kind of sticking out of the grass a little bit, I've seen it before, and let me dig here a little bit, because the last few times we saw something like this, there was something amazing underneath. And I'm not telling you how to build the roads, right? I'm just saying that, you know, let me do this thing here, and if we're lucky, we might discover something new, and you know, maybe we might discover some, you know, crazy network of underground tunnels that will also change how we build roads. But that is, I don't know that, and that is kind of separate. I'm interested in what's down there under this little thing sticking out. And to see why this kind of argument has some credibility, let me start by reminding you just how incredibly successful physics has been indeed. And let's discuss a little bit kind of why. So I start with this first point of my plan, and I'll start back in the 1800s with my first example that concerns these two gentlemen. So in 1821, people noticed that the observed trajectories of the planet Uranus disagree with predictions based on Newton's laws. So people calculated how the planets should be moving, and they found discrepancies. And physicists were so confident in their laws that instead of saying, oh, maybe something's wrong with the equations we're using, they instead said, maybe there's a new planet that we haven't seen before. And in 1846, this planet was discovered, in fact, within 1 degree of where it was predicted to be at that time. And this is the planet Neptune, of which we now have this beautiful picture taken by Voyager 2. So that's my first story back from 1800s. Let me tell you a more modern example. The Higgs boson that made a big splash in the news a few years ago. So this here is it's called the CMS experiment at CERN in Geneva. And it was built to test the standard model of particle physics. And one spectacular success of a few years ago was the discovery of this Higgs boson particle that theory predicted. So there was this press release that we observed, this, you know, this is in the middle of the detector. These are trajectories of the particles, which we can identify. And this is the event that, okay. So spectacular success celebrated by the Nobel Prize in 2013. Now here's an interesting fact. So physicists presumably were ecstatic. Well, they kind of were, but they were also, can you believe it, disappointed. Because with many people, the sentiment was, well, yeah, but we knew about that one. We were hoping for something new. And this is mind-blowing, because physicists were so confident about their theory, they were sure that this particle, this new form of matter, or actually new form of interaction that no one has ever seen, had to be there, to the point of being like, duh, where we actually found it, right? How arrogant can one get, right? And yet they were right. That prediction was correct. And this certainty about things that we can't possibly see extends far beyond these examples, and it's kind of become so commonplace that often physicists are kind of the first to forget how insane this is. And that's why I kind of wanted to start there, because here's another slide. This is here the history of the universe. From, you know, the Big Bang down here, and this describes the timeline of things that we know kind of happened in this order that they happened. And I don't know if you can see it necessarily on your screen, like here's a little thing that says 3 minutes after this Big Bang event, this happened. Nucleosynthesis and, you know, of light elements such as helium and lithium. 3 minutes, you know, 13.7 billion years ago, this thing happened, and then 3 minutes later, this other thing happened. How can we know this, right? And I was in a public talk where the speaker I don't remember where it was, but I remember a speaker talking about something like this, about this kind of amazing results from cosmology and, maybe black holes or something like that. And somebody from the audience asked, like, wait, you show these numbers, but, how can we know this? Are these, is this artist's impression? Are these guesses? Are we, and I remember the speaker replying like, well, yes, you're right. There's some sources of uncertainty on this. Like there's one source of uncertainty coming from here, then from there. So there's some error bars on this. And which is all true. But the first answer there is yes, we know it. And yes, this is absolutely insane. And yes, there's also error bars. It might not have been exactly 3 minutes. It may have been 3 minutes and 2 seconds. And we can bound it between this and that. And this just makes it more insane. So, okay, and as my final example, here's about this kind of the precision that people can achieve. Here's this. This is a famous example. Maybe you've seen this before if you've been in a physics talk of this kind of flavor. There's some property of a particle called the electron, which is called, for various reasons, anomalous magnetic moment. It's something that is measurable. It's a real property, and it has been measured. In fact, it's been measured, physicists call it alpha, and it's been measured to a ridiculous precision. This is the result of the measurement. This property of the universe, alpha, has this value, 15965, right? And this thing at the end, that's 73 plus or minus 28 on the 73. That's our best estimate of the value of this property. Now, why on earth would anybody think of measuring something to this ridiculous precision? Well, because we also have a theory that predicts the value of alpha. And here's what the theory predicts. And it predicts all these digits up until some discrepancy down there, which is within error bars. And just to give you some idea of what precision we're talking about here, this is 1 in 10 to the 13, so 1 with 13 zeros after it. And just to give you some sense of scale of how ridiculous that is, so, this is St. Louis, scale about 10 kilometers across on this map. And I zoom in on the Central Park. I zoom in on a leaf. I zoom in on that leaf. There's this, bacterium. Now I can't zoom in anymore because actually we get to the lower than the wavelength of light that we use to image things in our microscopes. And we get down to proteins, which we now draw as these cartoons because you can't image it directly. We use other ways of measurement. And this precision that I'm talking about is to say that for this protein that we predicted had to be on this specific cell and this specific leaf in this park in St. Louis, well, actually it wasn't here, it was there. And if it's not there, we sound alarm bells and saying, oh my God, there's no new physics, because we're so confident in these laws. So in fact, we just found, there's been a result that physicists have found a discrepancy in magnetic moment of another particle, and there's some excitement about that, and that is of a similar level of precision. Maybe you heard in the news, the anomalous magnetic moment of muon. So, okay. What I'm trying to say, though, is 2 things. So I reminded you of this, incredible success of physics, but there's something that I personally find more impressive than that in this history. The precision of 1 in, trillions of a prediction, that's amazing. But here's something much more impressive. The number of things, of foundational beliefs, that we never dreamt of questioning until we discover that they are wrong through this process. So here are some things. An object can be here or there, but not both, right? Space and time are not the same thing, right? These are not assumptions. These are things that are obviously true, right? Well, wrong, says quantum mechanics, to question 2.1. Wrong, says relativity theory, 2.2. And you know what? the GPS, the reason our phones can tell us where we are, the GPS in our phones, is because the electronics in these phones are built using the quantum circuits using kind of quantum effects, and the satellites on orbit, the GPS satellites that allow the positioning system to work, take into account relativistic corrections. So not only these are things that are wrong in some abstract esoteric sense, this is what the universe is like. And in our universe, these statements that seem so obvious are actually incorrect. And so, however impressive it is, as impressive as it is to predict something with this crazy accuracy of 1 in trillions, the more impressive thing is that we can somehow understand, make predictions, and reason in terms that are beyond any intuitive comprehension. So how did physics get there? We barely, for thousands of years, we barely made it to Earth being actually round and not flat, and then suddenly we're talking about curved space-time and these mind-boggling terms that defy any intuition, right? How did we get there? So I think for me, the methodology of theoretical physics has these two key ingredients that are responsible for its success. And let me point them out before I move to this, make this bridge to the context of living matter in biology. So the first key ingredient is that as a history of the discipline, we learned the value of imagining and studying worlds other than our own. And the second is that we learned to use mathematics as a crutch to go beyond the confines of our intuition. And so, kind of putting them together, one could maybe say that theoretical physics is science fiction written in the language of mathematics. And I certainly don't mean it badly, because it has something to show for itself. It has some successes that justify the approach. But sometimes engaging in science fiction is a valuable exercise. So let me comment on these two ingredients. So first, this imagining and studying worlds other than our own. This is referring to this kind of notion of generalization as the key tool that we use in theoretical physics to kind of understand and probe the world. And it's often levied as a criticism at physicists because, there's this kind of joke of physicists studying spherical cows in vacuum, right? Things that don't exist. But it's not a flight of fancy or a luxury. It's a deeply embedded part of the process, and I would argue it's the key to the success of the enterprise. And this idea, in fact, is very familiar in other contexts, which is that to understand an object, we should study the common properties of a family of objects. Instead of studying something directly, we should embed it in a family of similar things, and that gives us a context in which to study it. Now, this is nothing actually special to physics. So, think about the Iliad. How do I study the Iliad? Well, you can study it in the context of texts in ancient Greek. You can think of it as in the context of, epic poems in different civilizations. You can think of it as a representative of the tradition of oral poetry. There are other ways, right? And each of these contexts, thinking of Iliad as the example of this or of that or of that, gives you a different framing, a more precise question, gives you something to compare and contrast to understand what's in common, what's different, right? So to study the object, you embed it in a family of similar objects. Now, what do we do when our object of study is our universe? So our universe, and let's take it from some kind of early 20th century, kind of the turn of the 20th century picture of our universe, which has electromagnetic field, electromagnetic interactions. It has Newton's gravity. It has, we live in a three-dimensional space. We know some particles that exist. We know protons, neutrons, we know electrons. So how do we study this object, our universe? What sort of families of objects can we embed it in to do our, the compare and contrasting that we can do in science? Well, we only have one universe. And so we embed it into imagined possible universes, right? So instead of studying, specific circuits of electromagnetism, we could ask what different field theories exist. And that's a term from physics. It's a branch of physics that studies different possible interactions mediated by something like an electromagnetic field, but something that doesn't have to be the electromagnetic field as we know it, right? What else could it be? We live in 3 dimensions, but what would physics look like in different dimensions? What other forms of matter could exist, and what would be the mathematical description of that? And how do we know, if we knew how it would look, how that universe would look, then we could know, how can we tell that our world is not that one, right? How would we know that this is the only forms of matter? And in fact, they're not. We have discovered all sorts of other forms of matter, different elementary particles, right? And okay, so this is kind of why, and field theories, thinking about that is what gives you ultimately things like the prediction of the Higgs boson that we know existed. Thinking about physics in different dimensions, well, that surely is silly because we're in 3 dimensions and not 15. Well, for example, 2-dimensional physics is an incredibly now active area of also applied study with things like 2-dimensional materials like graphene, or it's a very active area also here at WashU. So that's partially why I use this kind of metaphor of science, comparison, analogy to science fiction, because science fiction can help us understand the role of some element of our society by imagining what a world might look like without, right? There is a similar kind of thought process you could imagine there. But there's an important difference. When we write science fiction, we're limited by our limited imagination. And some people have very active imagination and they can write amazing science fiction, right? But we have nothing to guide us. And so the key here, the key second ingredient is mathematics. So we use mathematics to write our science fictional scenarios and to study them. And so this is my kind of second key ingredient that I want to point out. So theories in physics, such as general relativity, the curved space-time, right, or quantum mechanics, they're famously very non-intuitive. And that's something that, is very present. Everybody kind of, even people who don't know anything about quantum mechanics know that it's something very non-intuitive. But here's a very important point. Well, of course they're non-intuitive. They had to be non-intuitive. Because why would they be intuitive? Our intuition developed at the scale of our world of meters, feet, miles. It has, our intuition is there to help us spot, to tell us when it's a wind rustling in the bushes or if it's a predator, right? That intuition has no business working at scales of which we have no direct experience, such as scales of galaxies or scales of atoms, right? And so our intuition, our expectation, or even the words that we use, that we develop to describe reality around us, do not have no business applying at scales so vastly different as the scale of our, Milky Way, or the scale of atoms, or, and this is where I'm making my bridge, or the scales of microorganisms. Because here's another world of which we have no direct sensory experience. the world of microbes is, when I go to a forest and I look around me and I ask, how do I describe what I see? Well, I see foxes, I see rabbits, I see oak trees. And so I say, well, so it's a natural language in which to describe the system is how many foxes, how many rabbits, how many oak trees? Do foxes eat rabbits or do foxes eat trees, right? And let me build my understanding that way. Now, how do we know whether that language is appropriate for microbes? And so what I'll argue for in the second half of this talk is that maybe microbial ecology, this world of microbes, might actually belong in this list of contexts where our intuition is beginning to fail us, and we might need to change some of our deep-held beliefs. Okay. So I'm transitioning to the biological part. And so, like I argued, our picture of ecology and evolution was shaped by this macroscopic intuition. And what does it mean? What sort of, remember, the point is there's some things that we don't even recognize as assumptions because they're so obvious to us. Well, of course it's like that. How else could it possibly be? But now, with this long history of seeing such postulates overturned in physics, now we learn to be sensitive, to be cautious, and to try and identify such assumptions. So, if I'm looking at a cat, it seems very natural that I can draw a bubble around a cat such that what is inside of the bubble is what I call my organism. What's outside is what I call its environment, right? It's a very natural thing. We don't recognize it as an assumption. Now, what this bubble actually kind of means technically is that what's inside the bubble changes slowly, and that's what we describe with evolution. So, cats slowly evolve, lions and tigers are different forms of a, common ancestor cat that sort of slowly transformed how they navigate their world. And the environment is a rapid change. Today there's a lot of mice around, tomorrow there's not a lot of mice around, and the cat has to deal with it. And that is ecology. And this assumed separation is fundamental to how we think about ecology and evolution and kind of just the language of life sciences. Because it's this separation that presupposes, for example, that is required for the, the central object of evolutionary theory, which is this notion of fitness, that organisms evolve to improve. Because fitness of what? Of the organism in its environment, right? And if I can't draw the bubble, I don't know what to call fitness. Okay, so this is our picture, and organisms in these bubbles interact with organisms in other bubbles. And of course, that's, we look around, that's exactly what the world looks like around us. But the question is, well, how much of this picture remains if I think about bacteria? Let's say that I replace the cat by an E. coli. To what extent all of this still holds? And it turns out that in a completely non-speculative way, but in a way that everybody agrees on, there's lots of evidence that the familiar words that we're using are not very good at describing microbes. So let's take, for example, the word species. Now there's an issue. The classical kind of biological definition of species, at least one classical definition, is kind of has historically been a group of interbreeding organisms with fertile offspring. Okay, now that obviously doesn't apply to microbes because microbes do not mate. They do not have fertile offspring, they just divide. Now, moreover, microbes can exchange DNA with each other, as we're recently discovering. And so this picture of, the tree of life, as we all learned in sort of textbooks, of tree of descent, it's not actually true. It's not actually a tree. right? So, okay, this picture is kind of out. Let me give you some very concrete example. There's a famous study that said that if you take, so there's the bacterium we call E. coli. Now there's a study where they took from different databases, published genomes of E. coli, and compared 60 genomes of what everybody agreed was an E. coli. And they discovered that these 60 E. coli's only shared 20% of their genes. And for scale, you know, for between us and chimpanzees, it's like 99 point something identical genomes. So here, for bacteria like E. coli, any two E. coli can differ over half of their genome. And that is obviously a problem. So there's this is not an E. coli, this is a different bacterium, but this is just as an example of, if I look at different individuals of what nominally people would call the same species, and these are different individuals, these are rows in this matrix, and the columns are different genes that they carry. You can think of them as functions, or, you know, if these were big animals, just familiar animals, these could be traits like, you know, do they have sharp teeth, or do they have, you know, thick fur or not, you know, so things that are here, we just think about them as being encoded in the genes. And these are all the same species. And the way people try to describe this is, well, there's some genes that everybody shares, and then there are others that are sort of like in and out, and, you know, you could have them, you could not have them, but somehow that doesn't change the definition of the species. Okay, let me give you another example. And example of the word organism. So here, let me tell you a story. It's the story of this organism called Methanobacillus omelianski. That's a Latin name. So the story goes, there was a microbiologist, I think in 19th century. His name was Vasily Omeliansky. And he wanted to find an organism who could, like a bacterium that could metabolize ethanol. So he was kind of interested in metabolism of bacteria. So he fed, that was a long time ago, so experiments were run differently. He actually fed rabbits with liquor and tried to find in their poop bacteria who could grow on a medium with ethanol in it, which is like, you know, vodka. And so he found an organism. It was named after him, Methanobacillus omelianski. And it happily grew. It was named after him in 1936. And this organism, people were studying it in different labs across the world for 30 years. And then 30 years later, people discovered that it wasn't actually an organism at all. In fact, it was a consortium of two organisms, two species, as far from each other on evolutionary tree as we are from E. coli. Completely different organisms, but they depended on each other so much that for 30 years, we didn't notice that it was actually a consortium and not a single organism. And at the time, that made kind of a splash, like, oh, you know, we knew there was something kind of suspicious about this metabolism because it wasn't known a case that was the only organism that was known to metabolize ethanol directly into methane. And so people were that's why people were studying it. And they say, told you. Turns out it wasn't one organism, it was two. And what I find very interesting in this story is that for 30 years, nobody noticed. For all intents and purposes, it behaved like one organism. And so whether it's one or two, well, maybe that's the wrong question. So let me give you another example. You don't actually have to go to microbial scale to start asking questions like this. So this is a cool, these are ants. They're called leafcutter ants. So they carry these leaves, they cut out pieces, they carry them to their anthill. They can't actually eat them. What they do is they do agriculture. So they actually, in their anthill, they grow a fungus. They feed the leaves to the fungus. They clean the fungus from bacteria, and then they eat the fungus. So they're running agriculture. And in fact, this fungus is not found anywhere else except with these ants. If ants die, the fungus also immediately dies. It relies on the ants to feed it and to protect it from bacteria. The ants can't eat anything else, so they're only found together. And in fact, the queen ant, when she goes off to found a new colony, she has anatomically a pouch where she collects the spores of this fungus and takes them with her for the new colony. And so one can ask here, well, should we be calling them one organism or two? Again, right? And that's, people were fascinated by these questions in classical biology for a long time. And actually, like specifically the way it relates to this definition of an organism is something that like David Queller and Joan Strassman are here at WashU. They've written some very influential papers on that. But part of the reason people were so excited was because it seemed so different and so strange. But for microbes, when you think about microbes, we cannot avoid this issue. And if you think this is kind of, maybe a rare example, well, think again, because we ourselves actually, as we now know, are not organisms, but ecosystems. We're covered with microbes, we're filled with microbes that are very important for our health, that digest our food, that train our immune system. There's evidence that they can affect mood and be implicated in some disorders, including possibly, mental health. And so that is really kind of crazy to think about, because when we say me, what do we mean, right? So, okay, after all this, let me go back to this. So to what extent these words apply to microbes? And what we're discovering is that, well, first of all, the separation of slow change, rapid change doesn't work because microbes evolve fast. And so you can't really define these bubbles in the way that we would have liked. And if so, then all of this kind of crumbles, because now fitness of who, right? In what, if your environment keeps changing? What do we mean by the organism? And I want to take a bit of a moment here and emphasize one subtle point. So in this example of this Methanobacillus emiliansky, I told you, right? Like, oh, we thought it was one organism, turned out to be two. It is not a question of defining organisms better. What I'm kind of describing to you is this intriguing connection with kind of theoretical physics, thinking about challenging the assumptions. The point that I'm arguing for is not to definitively settle, to find some better, theoretical framework that will somehow help us settle this question. Is it actually one or is it actually two? And not even to somehow redefine organisms so that in this one case, we could say, oh, it's 1.3, whatever that means, right? The point is that it is possible that counting organisms, this question that seems so natural to us, might be in some sense, in some deep sense, the wrong question. The question is it possible that countable organisms are an accident of life at our macroscopic scale? And that we're using, when we're asking this question, we're using the wrong language. So this distinction is subtle and very deep. And I'm allowed to say this because this is not my idea. This is an idea of theoretical physics built by giants before me. But like, here are two particles colliding in an intuitive Newtonian physics picture. Which incidentally, okay, I say intuitive Newtonian physics, it wasn't intuitive to Aristotle. Okay, so Newtonian physics itself when it came about, was a revolution that transformed our intuitions in the same way, in much the same way as, quantum mechanics. But these days we're taught it in schools, and so we're so used to it. So I'm saying intuitive Newtonian picture. But okay, so in this intuitive Newtonian picture, two particles collide and interact and separate. Quantum mechanics that came in the early 20th century, it's this deeper understanding of the fundamental physics. It is not a method for redefining the particles of Newtonian physics somehow better or more accurately. It is, so you may have heard of particle wave duality or something like that, that elementary particles are somehow both particles and somehow simultaneously waves. What that is about is that quantum mechanics developed a mathematical language where it is rigorously true that during the interaction, the number of particles is not 1.3 or 1.7, it is actually not defined. And it constructed for us a language where it became possible for number of particles to not be defined, except when they are far from each other, non-interacting. And this is what made it possible, for example, to describe situations where you collide two particles and out come three particles. And remember, three elementary particles. It's not that one particle, was a rock separated into two. So in a similar way, here, what I'm arguing for is this kind of, the dream is to, or the sort of intriguing question is whether the boundary of an organism, when we're talking about microbes, might actually be undefinable. And so telling this story in words and pictures is profoundly limited because the story is written in the language of mathematics. And without mathematics, we are at the shaky limits of our intuition and imagination. And so, what I'm saying to you may sound, I don't know if I'm necessarily succeeding at conveying this idea, but intuitively, what we're talking about is, okay, imagine that this is a bacterium, and of course, if I'm looking at bacterium, it seems like I can, draw my bubble around that bacterium, and what's the problem? Well, imagine that this bacterium spends its entire life in direct contact with this other bacterium, without which it cannot survive without. It is fully dependent on it, and vice versa. They're only ever found together, right? Separate them and they die. And when they divide, one divides, the other divides, and they stay connected to each other, and they, right? Then for all intents and purposes, if we insist on defining the notion of an organism, maybe we should count them together. But then imagine that there's another bacterium, another species, which is there almost all of the time, right? This one is 100%, and this one 99.5%, and sometimes it's not there, but then two others are there, and they fulfill the same function, and again, if you take them away, these bugs cannot survive. And then 60% of the time, you also have these others, and this entire consortium can only survive anyway, only because there's this other big guy here that secretes something that they're eating, without which none of this would be viable, right? And then there's these others around and others around. Where in this picture do we draw our bubble? And okay, now the question, of course, is, should we care? So I argued for some intuition that there's this kind of intriguing question about some concepts deep down being not very well defined. Should we care? And the answer is not necessarily obvious because, okay, terms species and organisms that I described to you, how they're kind of, failing us, well, they're obviously useful. Right? It feels even silly to put it on a slide. It's obviously the case, right? And everything we're doing these days all the progress that biology has made is made using these terms. And so, yes, deep down, we have this problem, and the fact that we have these issues, they're universally recognized. The fact that you, like I described, is not actually an organism, you're both an organism and an ecosystem. The current body of ecological and evolutionary theory does not know how to conceptualize that in some pretty fundamental way. Like you have to, in biology, you have to decide if what you're talking about is an organism, in which case, you know, it has fitness and evolution is optimizing it, or it's an ecosystem, and ecosystems do no such thing. There's no such thing as a fitness of an ecosystem. But we are both organisms and ecosystems, and take our bacteria away and we're not very fit, right? So, okay, we all agree it is an issue, but maybe it's, you know, theoretically uncomfortable, but not an issue in practice. And indeed, it is currently not a big issue in practice. And so the answer to this is perhaps. Perhaps that's all there is to it. But perhaps not. And that's why I labeled this at the beginning as a dream, because last few times these alarm bells were ringing, we discovered something amazing. And so it's fun to think about what we might discover here. And I want to add here that this sort of failures of macroscopic intuition that I described to you are not, it's not just a purely intellectual exercise. It is also a very practical matter in things like health and disease. So, like I told you, if I go in a forest and I ask, how should I describe this ecosystem? I'm asking, well, okay, there are foxes, there are rabbits. Do I see foxes? Do I see rabbits, right? This is who populates the forest. So classical ecology, for the longest time, was formulating their questions in this way. Who is there? And the hope is that if we can understand the interactions between species, we could predict the communities, and in particular, for example, given that they affect our health, the communities of bacteria in us, we could, you know, understand what constitutes a healthy community in our guts, for example, right? We're told we should be eating yogurt because yogurt has good bacteria in it that should be in our you know, digestive tract and help our digestion. Okay, so who are these good bacteria? And there's this study from 2012 where people did exactly that. They went and sampled, you know, 100 healthy adults and asked which bacteria are present on their nose and the mouth and the teeth, right? And here are the results from, that's a figure from that study. And the thing you're supposed to take away from this is, so, you know, every group here is one place on the body. So for example, tip of the nose, okay? So they swabbed 100 people, their tip of the nose, and they looked at which bacteria were there. And a column here, the color, tells you what phylum of bacteria were found there. And so the noses, you know, many people were mostly this red phylum, but in others they were dominated by this yellow phylum. And, you know, in the stool, it was mostly this blue phylum, but also this gray phylum, right? And in some individuals, it was all blue, and other individuals, it was all gray. Now for scale, what is this phylum? Phylum is a taxonomic grouping of bacteria, and you can't directly compare it to animals, but for scale, all vertebrates are a phylum. And so this difference here is kind of like I go in a forest and ask, what do I see? And I see a forest of trees. And then I go in another forest, and it's all made of snails. I don't know, like completely different. It's not like, you know, is it rabbit with gray ears or white ears? It's just something entirely different. And so what are we hoping to predict here? Given that, you know, all these healthy people are completely different. So no, we don't actually know what bacteria to put into yogurt. There's this mysterious term dysbiosis, which means something's wrong with your bacteria which nobody knows what it actually means, despite how frequently you read about it. But then there was this encouraging observation, which is to say, if instead of looking who is there, you ask what they're doing, like some function. So I'm looking at my forest, and sometimes it's snails and sometimes it's trees, but okay, how much light is being absorbed? And it turns out that these same communities, in terms of function, are much more stable. So here it's trees, there it's snails, but they're green snails, and they also do photosynthesis. Okay, my analogy is not great, but that is very encouraging. So maybe depending on the variables we use, depending on the language we use to ask our questions, some questions may be more answerable, right? These functional composition of these communities may be more stable and predictable. And that's very exciting. It's a very encouraging observation. Well, of course, it turns out it's also not exactly right in some very deep ways. So for example, the career of Jeffrey Gordon, who's here at WashU over at the medical campus, who spent his career studying malnutrition in children. They have a study where, children in Bangladesh, they're eating the same diet, they live next to each other, right? But some of them are healthy and others are severely malnourished because the bacteria in their gut are different. And yes, so this function is not that stable. They're eating the same thing. Like, it makes sense. If we're all eating, a hamburger, the community in there, maybe it's snails, maybe it's trees, but they all digest the same kind of nutrients. Well, no, composition also does matter. So in what language should we be asking these questions to hope for predictive power? That is a very practical question that we are faced with. Okay, and here it comes back to this picture that I showed you before. So here, what I just described to you, and I tried to make this loop, that in this case, the problem is that same species can have different functions, and same function can be carried by different species, and we do not understand this relationship and its fluidity well enough, because this is what this individual two functions relationship looks like. I showed you this picture before. And we know this is real data. This is, what real life looks like. And we know that it doesn't match our theoretical perspectives that we currently have in the classical theory, such as, classical species. It would be this perspective, which would say that, okay, again, remember, the rows here are individuals, and we have individuals that are like this, and maybe these are foxes. And then there are individuals that are like that, and they're all alike, and those are rabbits. And then there are individuals like the third type, and these are wolves, right? This is not what this data looks like. Okay, I'm running short on time, so so this is one aspect, like thinking about this kind of data is one of the things we're doing. So this is another existing model, kind of a cartoon, which also doesn't match these data. And so given that we have this disconnect between our theory and data, something uncomfortable in the data that our language is not describing accurately, what other worlds can we imagine for how these matrices could be structured, right? And what would that mean for the behavior of these communities? And how do we know that our world is not one of them? So, this is one of the directions that we're doing in the lab. Do they look like this? Do they look like that? Okay. Well, here I'm skipping a little, but yeah, so this is one aspect of what we're working on. We also work on other things where, for example, we and also others have shown that ecological communities with hundreds of species, whatever that means, like in your gut, can behave very differently than those with only a few species, like foxes and rabbits. And all these directions are united by this umbrella theme of asking how our macroscopic intuition may not apply to microbes And so remember these two statements that I kind of started with on my physics part. An object can be here or there, but not both. Space and time are two very different things. These obvious truths that we ended up realizing were not actually true. Well, we can now add to this list. So, at our scale, here's something. It's either a cat or it's a dog, but it can be both. Yes, true for cats, but not true for microbes. And turns out that actually, most of life on our planet is microbial. That was a big slap to our egos, but it's true. If anything, we are the exception and not the microbes. So for most of living matter, this kind of statement is not true. And another one is organisms and ecosystems are not the same thing. Well, that is not even true for cats, because a cat is actually an ecosystem filled and covered with microbes that are very important for the cat. And so these statements here, we are at a stage where we know ways in which they fail to describe the real data. We're not at a stage where this part is kind of speculative. What's speculative is, well, for these claims, we have quantum mechanics and we have relativity theory already. And here we don't, and possibly maybe not yet, but maybe someday soon we will. And that's kind of exciting to think about as a, motivation of a research program. Okay, and with this, let me stop and thank everybody those are people in my group and my mentors. But for this talk specifically, I want to help thank these individuals, Andrei Losov and Evgeny Virodov, who have influenced me. All right, thank you. And if you have any questions, we can talk about those. Yes, I see a hand, Marty.
Yes, this is Marty Einig and Michael. Has your bubble inquiry, kind of simplifying it all, now in recess, or has it generated more paths for inquiry?
So what I described to you is the motivation and the dream that connects the different lines of what we do in my group. So very few we don't have a project in the group which is where we sit down and ask, how could we, if not organisms, then what, right? How do we describe the world differently, right? But this is the connecting thread. And for example, for the the case of structures of associations between taxonomic classification of bacteria and the functions they carry, right? That is through that motivation, we have some very specific things that we're doing there. We're working on data. We're using this kind of theoretical motivation to find and mathematical toy models to construct new things to be measured in data. So right now we have a lot of data, and often we're limited by what should we be measuring about it. And so in particular, in this case, we know what a well-clustered world looks like of individuals that form these very well-defined species. And we know that this is not exactly what the data looks like. But in how to quantify that, right? What should we measure on these real data tables to be able to address that question and say, in this context, things look more like species, and in this context, they look less like species, right? You can only make it a quantitative question if you have something to compare to, right? So hope that answers your question. But a lot of what I talked about here is not specifically, you may have noticed, is not exactly specifically what I'm working on. This is more the the dream of why I think theoretical physics may have something to contribute to this recent excitement about learning more about microbes, and why it's a kind of very productive area for overlap between physics and life sciences.
May I add that other pure scientists in the past had to create another branch of mathematics, such as calculus, to describe their intuition. Perhaps do you see that maybe you're going to be called to that kind of task?
So that's actually a very deep observation that you just made, because actually, so when I teach mechanics, we start the class by asking, well, what's so difficult about mechanics? Like, how difficult was it to notice that F is ma, right? And what's difficult partly is that Newton had to invent the whole notion of acceleration to make that statement. So he had to invent that mathematics. That is exactly right. And it is, I don't know how to solve that, but it is a deep issue here, because biology is all about hierarchical structures. So, and we don't have naturally hierarchical mathematics for describing this. So the way we know how to deal with this is to kind of cut off that hierarchy somewhere. For example, say, let's talk about, back to like, species, right? There's some tree, which is not actually tree, as I told you, but whatever, it's still, there's some hierarchy, and we can only think about it in terms of these like well-defined boxes. And we know that they're related to each other in this hierarchical way, but we don't have a natural hierarchical language to describe this. So we can choose a different level, and we have to try to be aware that we could have chosen a different level of description, but we don't have good ways of naturally capturing that, of writing equations in some hierarchical way, whatever that means. And so it's this situation where if somebody invented that kind of mathematics, I think we would recognize it as, that was the piece that it was missing. But how do you go about that? That is a very exciting question. And yes, I do believe that it is part of the challenge.
Very good, thank you.
Thank you for your question. I see questions from, I'm not sure who was first, Jeanette, or maybe let's, Jeanette, I see you at the top.
Yes, can you hear me?
Yes,
Yeah, it's actually Jay. I'm with Jeanette. So do you do experiments, actual physical measurement experiments?
No, I do not. Myself, I'm a theorist. I work with experimentalists, mostly actually experimentalists who run kind of biology labs, not kind of physical labs. So experimental evolution, for example, but I do not do experiments myself.
So in understanding the types of experiments that have to evolve this theory or the application of physics and mathematics to biology, do you suggest to the experimentalists what measurands they should be looking for in trying to determine what's going on in a given ecological environment, different from what they have been measuring?
That's an excellent question. That is the hope. So right now we have enough data that things like this is just a little snippet. We have massive databases where there's we amassed knowledge that exists, where we can start asking these questions using data that already does exist. But eventually, of course, so the hope is to identify new things to measure first on the data we already have, but then to refine the questions enough that we can actually, we'll understand what it is that we have not been measuring. Like, for example, right now, a lot of the measurements that are happening, we're sequencing DNA, and largely it is because we learned to measure it. And so in a way, it's this curious case of looking under a lamppost. We postulated that that's the thing that matters, partially because that's the thing we learned how to read. And it's a huge industry these days but it's partially because we have no, clear incentive of, developing the technology to measure something very specific that's different, right? There's already such a wealth of knowledge in those DNA sequences that we already don't know how to interpret. So yes, I'm not sure that was a very good answer, because
No,
ultimately I just said,
I understand.
yes, that's the hope.
So I guess what you do is after you analyze the data you've got, you'll determine whether it adequately explains or connects the cause and effect relationships that you think that we all thought existed before. And if not, you'll hypothecate some new experiments that should be run.
Yeah, and like one semi-sharp way of trying to ask this is like, well, we know that we can describe, like, for example, these like gut communities. That's what I briefly alluded to at the end. We can describe them compositionally, who's there. We can describe them in terms of this representation of functions. It's kind of like overall in this forest, how much grass is being consumed. And I kind of don't care if it's being consumed by, locusts or, I don't know, rabbits, right? Which so the thing is that ultimately, to be able to predict everything, we need to know everything, and we'll never know everything. But for every specific question that we want to ask, there is the right variables to measure to help answer it. And this mapping from the question to the right things to measure is something that we're just kind of building as a discipline. And here by a discipline, I mean kind of the collective we of microbial ecology and the kind of the biology side. They're thinking about this, in different ways. But yes, we're all kind of sharing that dream and try to bring whatever tools we have in our training to hope and make progress on that.
So in a sense, you're constructing some mathematical models that are much more complicated and inclusive than the biologists had used before, using physical mathematics or physics mathematics, tensor algebra, and so on?
Actually, no. It's actually usually the opposite, which is that physicist models tend to isolate one phenomenon to focus on, and then make a very simple model, much simpler than typical models in biology. The hope is, so that's this kind of ingredient of imagining the worlds. So for example, in the case of thinking about these data, we know we're treating these kind of mapping from species to function as if the world were well clustered, and there was this species and this is what it does, and then there's that species and that is what that does. So this is a model for these data, which is kind of this well clustered model. Now we can imagine and kind of dream, and let me try to walk you through this argument and ask, could we imagine a world with no species whatsoever? Now this is not our world. Our world does not look like that. But can we imagine how an ecosystem would behave if the association between, species and functions were completely unstructured? And we believe, okay, this is an imagined world, right? But if we eliminate from the whole, by construction, you cannot ask familiar questions like how many species are there, right? Because you eliminated, there's no species, right? So, but it's still a well-defined, like in this imagined world, it's a well-defined ecosystem. It's processing nutrients, something is happening, evolution is going on, right? It has well-behaved dynamics, but the question is, well, we have to come up with new terms in which to describe these dynamics, because familiar questions by construction don't work, right? So then, and if we came up with a description for these dynamics, then this exercise forced us into constructing new measurable quantities, which we can now go back and take to our world and ask, well, how do those observables behave? Those observables which, thanks to this mental exercise, do not depend on how well-defined species are. And so, the temptation, we that way sidestep the temptation of continuing to use this language, which we know doesn't quite work very well, but it works quite well. And so why would we go beyond that, right? I hope that maybe it's a kind of it's the kind of exercise that's familiar in theoretical physics elsewhere, which we're trying to bring here to this set of questions.
It sounds to me like you're trying to generalize from a many-body problem down to a two-body problem or something like that, and see if you can figure out whether you've correctly done that.
Yeah, so for example, you can have a many-body problem of Newton-type particles, and you say, well, but given that particles aren't very well defined, and it seems like maybe we have to describe them in some wave-like way, well, how do we do that? Well, let's simplify that from many bodies to just two, but try, or maybe even one, right? And try to understand what is this wave-like nature of a particle, even though in real life there are 17 particles there, right? But it's very hard to think about 17 wave-like particles. But if we think down to, let's just say two that are interacting, then this is simple enough that we can generalize and then go back to the original question. I think that's probably a good mapping there. Thanks for.
I have a question that is kind of related with that. But before that, let me congratulate you for one of the most interesting and motivating talks in outreach series I ever heard. Okay?
Oh, thank you.
Even in the physics part that I know a little bit more. I know nothing about the biology. My original question would have been if you can relate, when you presented the ecosystem with all the bacteria, that all the time you have different combinations of possible combinations of interactions, if you can somehow list all the interactions. And then my question was evolving according to your answer, your previous answers. One of the big differences that you have with physics is that electrons are all electrons, they're all the same. We count them as all the same. These bacteria, not even of the same species, are not the same. So can you ever list anything, or can you ever classify what you are seeing? And can you even interact and say, okay, I take this bacteria and put a different one, let's see what happens? That is possibly my question.
That is an excellent question. And well, so you kind of put your finger on a big issue, which both has multiple facets. So one is, you could take that as an example and say, here's why this whole hope of bringing methods of, any sort of analogies of, oh, in physics, we think about a gas and there's all these particles interacting, and yet, see, we managed to predict something, and therefore let's try and do the same thing in biology. And that's the argument of, no, it's not going to work like that, because in physics, your gas was, all these molecules are exactly identical to each other, and they didn't want anything. They didn't evolve to, hit this piston as many times as possible because their life depends on it, right? And biology is that, and therefore these methods will not work, right? On the other hand, you could also say that this is kind of turn it more hopefully and say, this is identifiably like a challenge of describing the systems. And if we could develop, for example, mathematical language that could somehow support this hierarchical nature of description, right? So imagine that I'm describing my organisms by some, long list of traits, and somehow in my theory, it's allowed, or like that this length of that list is in principle infinite, right? I can always look in a bigger magnifying glass and distinguish yet another characteristic and add it to the list. And somehow the equations that we need to be thinking about need to be compatible with this fact of biological reality, right? Which constrains the kind of mathematics we can do. Because if it's not compatible, we know that that's not the fundamental description of the system, right? And that actually constrains the search for these kind of mathematics. I'm talking about this from this angle because like a recent thing that I'm very excited to kind of direction in the group is a way to think about ecology in a way that's compatible with making the list more and more kind of longer and longer, and say that in principle, there's these finer and finer distinctions that interact weaker and weaker with other things. And you can actually see that there is a regime where having your list too short completely changes your predictions, right? Or you could have a regime where if I'm only interested in these predictions for like, you know, this course level, it's fine to truncate my list here. If I want to predict more, I need to make my list longer, right? But developing these kind of mathematics in these toy worlds where, like what I'm just describing to you, this exciting direction recently, it's in a toy world where these kind of abstract organisms interact only by competing for, you know, nutrients. That's so, you know, real world is so much more than that. Bacteria also poke each other with, you know, weapons and secrete antibiotics that kill things, and they, you know, benefit from protection from bigger organisms. There's so much, right? And so in a sort of any sort of sensible biological model there would try to include all that complexity. And that is why there was this excellent question before, and kind of the physicist approach here is to say, well, but we're trying to nail this structural problem. So let's simplify away those other factors and zero in on this fundamental issue. And then we'll try to reintegrate the complexity later, which also means that then it is pretty hard to compare it to data directly, right? And that's why I had this long tirade before about, well, we need to figure out what to measure, because of course our models are much too simple. So we need to understand what is the thing that would distinguish predictions from this model of predictions that, you know, did not incorporate those, you know, ideas in the face of everything was simplified away. So yes, that's the name of the game. That's why it's exciting. That's also why it's a kind of,
yeah. On top of which it sounds like everything is non-linear. So you cannot really simplify and leave something linear, but then you cannot analyze it very well if it is non-linear.
True. And a lot of what we do is in simulations because we don't want to simplify away the nonlinearity. So we kind of, yes, that is another concern. Yes.
Very interesting. Thank you.
Thank you for the excellent question.
Mikhail, Mikhail,
Yes.
John Burns had a question, which he's been waiting patiently to hear the answer to.
Oh, sorry.
And it's a deep question. Are genes always made up of the same limited library of particles?
Yeah, sorry, this was in the chat and I didn't see the chat. Well, I guess there are many ways in which to take that question, and maybe I'm not entirely sure whether my answer to it will be how you meant it, but well, on some fundamental level, you know, yes, because it's, you know, A, T, C, and G, but that's not the level which we're describing here. These are AT, you know, the molecular code of the DNA, but that's certainly not the level how we're trying to describe it here. There is maybe one thing that I can comment on here is that what I was describing as kind of functions are certainly not encoded in individual single genes. And that is one of these layers of complexity that I referred to in terms of trying to compare this to data, because the data that we have that are good data are about, you know, was this gene present or absent in this genome? Now, the mapping from genes to functions is complicated. We don't always know how to assign all genes to functions. There's also no such way of asking, was this same gene present, this gene that I saw in E. coli, was it present in this other species? Because in this other species, even if they have a close analog of something doing exactly that, it won't be exactly that gene. It'll be quite different. So hopefully that, I feel like I said a few things that are next to your question as opposed to answering your question, because I'm not sure what you mean by particles here. If you can clarify, I can say more.
Can you hear me?
Yes.
Okay, so I was wondering that thinking of physics, there's the classic particles. So in genes, um, are there just these classic particles? Do you ever see anything different than these? I don't know. These are acids. Are they proteins? Because this seems to be the fundamental building blocks of whatever we're calling these organisms. It seems to be a starting point, and then you make the leap to functionality, which is there's a miracle occurs, if you've seen the joke.
Yes,
Between
yes.
man. So that was, you've kind of answered my question.
Yeah, okay. And then I'll just maybe add one thing here, which is that often the question the big part of the big question is what's the correct level of description, right? And in fact, what I described to you, for example, and there's like, oh, organisms are being maybe poorly defined. It's kind of it's an example of that question, right? What is the relevant unit of interacting things? This is far beyond, this one area. Like in neuroscience, people are discussing whether, for the longest time, we're thinking of neurons in the brain as a neuron is the unit of computation. So how would you be describing the brain to understand how it's performing computations? Well, it's a bunch of interacting neurons, right? Now that's actually not obvious, because it could be that, a neuron is a cell has a lot of dendrites. There could be computations happening at the level of individual dendrites, and they're far away, and yes, they're kind of fed by the same cell body, but maybe the unit of computation is much smaller than that, right? Or maybe, so here in this ecological context or evolutionary context, should we, we all carry, our cells have mitochondria, which are these kind of energy, power plants of the cells. You may know that mitochondria used to be free living bacteria. And actually they still have DNA, all of our mitochondria. And if you read the DNA, they are identifiably similar to their current free living relatives, right? Now, we're not thinking of ourselves as constantly at that level of symbiosis with these bacteria. By now, they're so deeply embedded in us that's ourselves, right? But then, but then there are these other bacteria that live on top of us, and how should we treat those, and blah, 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 right? So it's all this question of degree of the level of description. And so in particular, everything I was talking about today was about level of description, basically cell or above. Now then there's, people like, Dawkins or whatever, selfish genes, is whether maybe genes are the relevant level. These kind of go below the cells, right? When you're thinking about cells or above, I'm not even thinking about exactly how it's encoded in these, proteins, although the proteins are the things that are functional, right? For everything that I was talking about, that we're not even touching that, because it's, it's, it's a few levels of description down, and it's, yes, we have to pick our battles, but that's a great question. How is physics changing as a result of your work and that of others, and what comes next? Ooh, that's a great question. Well, that was the thing, this series being called The Future of Physics, which is partly why you may have noticed I hardly talked about what I do, because that seems preposterous, and instead I described to you my kind of imagined picture of the future of physics, kind of from my vantage point and my interests. Although yes, this is kind of the area I'm working on, but okay, how exactly what we've done so far is changing physics. That is more of a long-term career prospect. We did get to contribute somewhat to this study, kind of this revival of bringing methods of statistical physics to study ecosystems with a large number of species. Like I think I briefly mentioned that, you can show, like in physics, we study phase transitions, and phase transitions are about the fact that when, you study interaction of 2 particles, 3 particles, and you think you understood something, but turns out that if you have tons of such particles, suddenly the behavior is completely different, right? And that's a very interesting idea for, could something like that be happening for, for example, ecosystems where we understand, we put in our petri dish 2 bacteria interacting, we think we understood something, and then we try to extrapolate to real world situations, but in our guts, there are hundreds and hundreds and hundreds of species. And maybe their behavior is completely different in a different phase, different regime, right? So there's been some classical work on ecology there, and we kind of brought some methods from what's called disordered statistical physics to study this in the context of this models of competing for resources as a kind of proof of principle that something very different could happen, indeed. You know, there's an area of physics of kind of this, how is this changing physics? Well, a lot of statistical physicists are interested in this context for reasons that somebody mentioned. You know, we're thinking about particles that are, in this case, kind of all different. And, you know, is there new ways of, you know, thinking back about physics questions? And like there's a, yeah, so, okay, let me cut that there and say, I'll come back in 10 years and tell you what the influence was on the on physics of what we did. All right.
Hello, Mikhail.
Yes.
So I have a dozen questions I could ask you, but I'll just focus it on one to keep it short. I've always wondered, okay, so if you send an astronaut off to Mars and we colonize Mars, can people, you know, how the microbes in their guts and stuff might change? What kind of microbes would they use? Also, if you put a person with immunity issues and you put them in a bubble, okay, could people live without their microbes? I think the answer is no, but would it have benefits? Would there be problems? What kind of microbes would you have to take with you to go to Mars? You know, I'm sure that these are questions that you're probably thinking about, but I've always wondered what would happen if we tried to live without them.
That's a great question. So turns out that we do actually know something about that a little bit. We collectively, I wasn't ever working on things like that, but I did work actually. Well, no, actually, I was even involved in a study that is even related to your question. Not in Mars and not in humans, but in mice and in a lab. But so we can't test what would happen to humans in such bubbles. They don't let us run experiments like that. But we can do this with mice, and people do that a lot because that's, you know, it's key to asking questions like this, to have a kind of model system. And mice are actually reasonably happy. So when mice are born, they're sterile, and then they're, you know, inoculated with bacteria from their, you know, their mother. And if it's very hard, but if you keep them in enclosed things and you sterilize the food you feed them, and you only handle them through this kind of, you know, glove box thing, and are very careful about this, it's possible to keep them what's called gnotobiotic, without their bacteria. Or germ-free mice. Gnotobiotic is when you then inoculate them with some specific bacterium that is you're interested in or something. So they are reasonably healthy, depending on different, like, you know, in that box, they also don't encounter a pathogen, right? Or things like that. So, and different animals survive differently without the bacteria. So it's not true that we cannot survive without them. It is, what effect it has on physiology of the organism, of the host, is a very active area. People actually are studying this. I'm sure that NASA is funding some research in the astrobiology kind of, or, you know, Mars direction that actually is addressing those questions for those reasons, as opposed to just for fundamental science health reasons. Because like I mentioned, you know, you sample healthy individuals, they're all different. So how do you make any sense of anything? You need reproducible experiments, right? You need some replicates. So how do you do that? You take a germ-free mouse, inoculated with what's called humanized mice. It's not the terrible thing that it maybe sounds like. It's just they're given some bacteria from humans, so their gut composition is kind of similar to that of humans, and then you see what. And so one of the study I was involved with is that if you do that in this kind of germ-free setting, they actually kind of, depending on the diet, they lose some strains from the gut. And their ability, so fiber is a healthy component of a healthy diet. If you feed your mice low fiber diet, they will progressively lose the strains from their guts that help them digest the diet, the fiber. And so what this means is that then to recover that ability, it's not enough to just start feeding them healthy enough again. You also need to introduce the taxa that they lost. And the study, which this was a part, actually kind of suggested, the pitch was that we collectively as a culture kept eating, what's called westernized diet, which is not particularly healthy, for long enough that maybe we as a culture may have lost some of those strains that, would have been part of our healthy digestive tract, that dietary interventions might not be enough. We also need to reintroduce some of these lost taxa that indigenous populations have preserved. So that's kind of a little tangent to something real life, but yeah, thanks for the question.
Okay, it looks like you've answered all the questions for now. Remember, you've just promised to come back in 10 years, so I'm going to put you on the schedule.
Excellent. I'll put it on my calendar. Thank you so much, everybody. Thank you for great questions and for coming out on a Saturday morning. And I'll see you at the next Saturday Science Series lecture.
Thanks a lot.
Have a great day.
Okay.